Hello and welcome to Taconic's webinar entitled Microbial Monitoring of Germ-Free Isolators, presented by Paula Roche. Please allow me to introduce myself. My name is Margit Wissenbach and I will be your moderator today. Before we get started, I'd like to address a few practical matters. This webinar will be made available to all attendees. We will email a link to the presentation to you by the end of this week. Directly after the presentation, Paula will answer your questions as they appear in the control panel. So if you have a question, please type it in the control panel. The other attendees will not be able to see a question as it is sent directly to Paula and me. If you cannot see the control panel, just single-click on the single arrow on the top of your screen. In case your question is not answered at the end of today's session, Paula will respond to you within the next 24 hours via email. If you are experiencing technical difficulties, please send the host a chat message, and our staff will be able to assist you. So let's get started. We hope you will enjoy today's webinar, Microbial Monitoring of Germ-Free Isolators, presented by Paula Roche. Paula holds a PhD in microbiology and immunology from Wake Forest University in Winston-Salem and a Bachelor of Science in Biology from St. Andrew's Presbyterian College in Laurinburg. At present, Paula is Associate Director of Health Monitoring at Taconic, Rockville, Maryland. She is responsible for direction and oversight of the global health testing program performed at all Taconic production and service sites. Her responsibilities include review and improvement of health testing and provision of technical expertise to both management and technical staff at Taconic's diagnostic testing sites. This webinar will cover microbiological monitoring of germ-free isolators. The topics will include monitoring of newly set up of isolators, supplies, and testing post-animal entry. And with this, I'm happy to hand over the word to Paula Roche. Please, Paula.
Thank you, Margit, for the introduction. Good morning, everybody. Today, I'm going to share with you how we monitor our germ-free isolators at Taconic. What I'm going to cover will be applicable to both flexible film isolators and semi-rigid isolators. I've divided the presentation into multiple sections, and I can't seem to get the slide to go forward. There we go. I've divided the presentation into 4 sections. I'm going to start by sharing with you how we do our sterility testing of the newly set up germ-free isolator prior to animal entry. Then I'll talk to you a little bit about the quality control of materials that enter the isolator. Then microbial monitoring of the isolator after the animals are entered, and supply testing and how you may have to outsource this if you don't have a microbiology lab on site. So typically, when we set up a germ-free isolator, we already have the supplies. This includes water bottles, racks, caging, all that sort of thing already in the isolator, as well as feed and bedding. We also set up a system we call a mold trap, which essentially is feed and water, and we put this into an actual rat or mouse cage, depending on the animals in there. And we use this as sort of a living indicator of whether or not mold has gotten into the isolator. So we do 3 weeks of sterility testing prior to entering any animals into the isolator. During this 3 weeks of testing, the results must be negative for everything. So week 1 testing includes testing swabs that are placed into the mold trap. We also moisten swabs with sterile water and swab the interior walls, including the ceiling, the gloves, all of the filter outlets, any caging, feeders, as well as the floor area of the isolator. These samples are then sent to our quality control laboratory, and I'll talk to you a little bit more later about what we do with those. For week 2 testing, It's very much the same thing. Again, we take mold samples from the mold trap, and we take swabs and swab the interior of the isolator, caging, feeders, floor area, just as before. Week 3, the same thing. And as I said before, we have to have 3 solid weeks of completely negative results before animals are ever entered into the isolator. It's very, very important when you do this testing that you do not get sterilant onto the swabs. Obviously, if you have sterilant on the swab, all of your results are going to be negative, and you will not have an accurate assessment of the microbes in the isolator. So what happens to these samples? Well, these samples are then transported to our quality control laboratory, which happens to be on the same site. These samples are processed the same day they're received and collected. They're cultured both aerobically and anaerobically. We also prepare wet mounts of these samples. We keep the original testing vial for at least 1 week, and this is in case of benchtop contaminants and also for mold growth. Sometimes mold won't necessarily grow on the plates, microbial plates we use, but it will often grow within the vial itself. It often likes to grow on the wooden stick of the swabs, so it's very important to keep these vials. We also keep the vials in case of any discrepancies so that we can go back to the original testing vial. So what happens if you find something? So what we do is we must confirm a positive finding with a second sampling, and this second sampling has to be taken on a separate day. If it doesn't confirm, then an investigation will take place, and usually that involves talking with the staff in Notobiotics about sample collection. You know, could anything have gone wrong with the sample itself? We still must have 3 consecutive weeks of negative results for the isolator to be used. So if there is a positive finding, that means you must have 3 weeks after that of negative findings for the isolator to be used. So what about the things that go into the isolator? Well, at Taconic, we have a policy that for germ-free isolators, absolutely everything that goes in there must be autoclaved. And we can talk about this a little more later if there are questions. Obviously, depending on the kind of studies you're doing, you may have to bring things in that are not autoclavable. And there are some approaches you can use to test these things. But back to autoclaving. So one of the most important things you can do is be absolutely certain your autoclave is performing as it should be. I know particularly when I worked in an academic setting, we didn't really test our autoclaves on a regular basis. Here at Taconic, we validate all of our autoclave runs, and we have very particular validated cycles that we use for our germ-free isolators. In this case, certain amounts of things can only be entered for certain autoclave runs. And each one of the cylinders that will ultimately go to a germ-free isolator will have a biological indicator, and we'll talk a little more about these in a minute, and we'll also have chemical indicators within different portions of the cylinder itself. So you may also be using ethylene oxide for sterilization purposes. If you do, keep in mind there are also biological and chemical indicators available for those as well, and you will want to validate that for your particular run cycles. And again, as I mentioned, we generally do not admit anything into the isolator that's not autoclaved, but I do know we've had customer questions regarding irradiated feed and how could you use such a thing in a germ-free isolator. And so we can talk about that later if anyone has any particular questions. So what are biological indicators? I'm not recommending any particular brand. I just chose ones that we use. There are lots of brands of all of these types of indicators out there. One of our favorites is the Verify, which is produced by Steris. This is they term SCBI for a self-contained biological indicator. And what that means is it requires very minimal handling. There's actually a spore strip inside of the vial. You autoclave the entire vial, and then you can crush. There's a little media component in the bottom, and when you crush that, the media is released, and then you wait to see if there's any growth. Typically it's 48 hours, and if it's positive, the vial liquid turns blue, and if it's negative, it doesn't. Or vice versa. I think perhaps it turns yellow and it starts out blue. But in any case, it's a color indicator, very easy to read and very easy to use. Depending on the type of sterilization that you're doing, you'll want to make sure that you order an indicator suitable for that. They typically use different types of bacillus because bacillus is a spore former, and the spores are very difficult to kill, and so it makes a really good test of your system. There are other types of biological indicators besides Verify, as I mentioned. The ones shown on the left on this slide are made by another manufacturer, and they actually have different colors for different types of sterilization. And then on the right are spore strips themselves. And this is similar to what is inside of the self-contained biological indicator, but in this case it's separate. And so it allows you, it's just a little flat strip. This allows you to put it pretty much anywhere in the autoclave and anywhere in your packaging. The problem that we have had with this is keeping them sterile, because if they get damp on the outside of the paper, by the time you transport it to the lab and get it into culture media, it can sometimes be contaminated with environmental contaminants. And so I don't particularly care for using these, but some people prefer them. So chemical indicators are a lot cheaper than biological indicators, and so we tend to use a lot more of these. These are also nice because they're flat, and they're essentially a little flat paper, and you can put them inside of bags of feed, underneath the containers that you're autoclaving, inside of a cylinder that you might have full of supplies. You can tuck these in. We generally use several of these in every load. These have a little chemical bead at the bottom end of the indicator, and in the presence of the steam and the increased temperature in the autoclave, the chemical bead melts and then migrates. As you can see, there's a reject and an accept portion on here. And what you want to make sure of is that it goes into the accept portion. I would not personally recommend using these alone in your loads. You should always try to include at least one biological indicator. They are much more of a challenge to sterilize. And so knowing the value of the animals you're going to place in your germ-free isolator, you want to be absolutely certain everything is sterile. So then, after we have our 3 weeks of negative testing results, animals are entered into the isolator, and we do weekly vial testing. And what this is comprised of is sort of similar to the testing that happens in the beginning. We have sterile swabs that are in the isolator, and these are in sterile vials, and they're entered as part of the supplies. And every week, one of the caretakers will take out some of these swabs, moisten it with, I have on here, dirty water, meaning water from water bottles that the animals are actually drinking from. So they moisten it with that water, then they swab the caging, the tops of the feed, the feeders, the floor area, everything in there. Then they randomly sample 10 to 12 cages. From these cages, they will remove fresh feces, a little bit of soiled bedding, and then they dump in a couple of milliliters of water from several of the water bottles that are in use. So you end up with this vial of this gamish of bedding and feces and swabs. This vial then is immediately transported to our quality control laboratory where it's cultured. And here below I've described for you the actual plates that we use when we're looking at these. So we generally plate these on trypticase soy agar with sheep's blood. This is a wide general growth media. Pretty much anything will grow on this. We use a nutrient agar with dextrose. This one we incubate at room temperature rather than 37 degrees, and this one tends to pick up more of the environmental contaminants. Then we also use Brucella agar with 5% horse blood. This is incubated anaerobically at 37 degrees. We then prepare wet mounts of some of the liquid in each of the vials, and those are examined microscopically for any signs of bacteria or mold. And then once again, we keep the original vial sample for at least 1 week so that we can go back to that should we need to, and also to monitor for mold growth. One of the things to keep in mind is that the things that we see typically contaminating the isolators are almost always spore formers. And this goes back to autoclaving and sterility monitoring and sterility testing. The spore formers, excuse me, are more difficult to kill and more likely to survive. And so you want to pay special attention to looking for those, particularly bacillus. Also for mold. So you want to really keep these plates for at least a week and keep checking them for mold growth. So I'm sure that some of you out there don't necessarily have a microbiology lab just down the hall where you can pop in and drop off your samples. So one of the things we've been looking at for our own use, and then also so we can share with our customers, is looking at the viability of these organisms in different transport medias. And this would allow you to collect your samples and then ship them to an outside diagnostic laboratory. And there are many of these all over the country. And I think at the end of my presentation, I do have a couple of websites, and I'd be happy to help you, find one closer to you. These different types of transport media aid in keeping both anaerobic and aerobic organisms alive. Generally, the companies say that the organisms will live for up to 72 hours at room temperature. They come in all different formats, as I've shown here on the right. There are tubes available, different types of jars, vials. There's one system called the BBL Protocol Transport System for fecal samples. And as I mentioned, we're in the process of validating these transport systems right now. And I would highly, highly recommend that before you use one, you actually do some testing of your own. We're doing this testing by comparing essentially RT equals 0. So what does it look like right when you collect the sample and you plate it versus 24 and 48 hours later, how the sample will grow after you've shipped it somewhere. And I have to say that so far we've had great success with this, and once we have it all validated, we'll be able to share more of this information. But I'm happy to tell you if you're interested in the systems that we found have worked the best. So in summary for my presentation today, we went over the microbial monitoring of the new germ-free isolators. And recall that I said you want to have at least 3 weeks of negative results prior to animal entry. One of the other key components is making absolutely certain that you do not have sterilant on the swabs when you do this collection. Otherwise, you could have false negative results. We also talked about the QC monitoring of the supplies. And as I said, we only allow autoclaved materials into our germ-free isolators, but sometimes you may have extenuating circumstances where you have to enter things that are not autoclaved. It's very, very important to validate your autoclave cycles. You need to validate your run times, your load sizes, and everything that you're autoclaving. I highly recommend the use of biological indicators in addition to the use of chemical indicators. And then after animals are entered into the isolator, we monitor the isolators weekly with vial testing. And the vial testing is comprised of swabbing the inside of the isolator, taking a fecal sample, water samples, and putting all that together and plating it and looking at what grows. So I know this is a little short, but this concludes my presentation, and I'm hoping that the attendees will have lots of good questions. Remember, your questions are all anonymous, and we are happy to answer anything you want to ask. So Margit, are there any questions?
Thank you, Paula, for this interesting presentation. So yes, our question and answer session has started, and there are questions. So the first question is, what if I have to use irradiated feed for my studies?
Okay, in the case of having to use irradiated feed, what I would recommend doing is actually sterility testing the feed. And what we do at Taconic, because we do use irradiated feed in our isolators, just not our germ-free isolators, we do sterility testing in our quality control laboratory. And what happens is the feed is placed into a biological safety cabinet, and we take samples from around the bag. So we essentially make sterile slits in the bag of feed, pull out pellets. These go into vials of testing media. And again, we use just a general growth media. In this case, it's like TSA broth. And we culture this for at least 48 hours. And if it looks like there's no turbidity in the vials, we will also plate these onto TSA agar just to be absolutely certain. And so we do this to monitor the sterility of the feed, and in all honesty, we have had failures where the vendor has told us the food is sterile and it's not. So you absolutely want to test this.
Okay, thank you. There is another question. I'll just take another random. What media do you use for aerobic and anaerobic cultures?
Okay, we for our aerobic cultures, as I mentioned, we use TSA with blood, which is a good general growth media for pretty much everything. And then we use another NAD media and grow that at room temperature, which will pick up most organisms that are environmental, which in a lot of cases that's actually what will contaminate a germ-free isolator. For anaerobic culture, we use the Brucella agar with horse blood. And again, when we do that, we check it at 24 and 48 hours.
Okay. Another question. Can you please share with us your autoclave cycles for both liquid and dry content?
Okay, for that, That's a little more complicated of of a question because the autoclaves that we use at Taconic are almost like a room size. It's you know, when I first came to the company, I was actually surprised at how large these are. You know, you can like drive a golf cart into them. So the cycles, I would recommend actually contacting whoever the manufacturer is of your autoclave and asking them for recommendations and then validating that the cycles actually work for the size of loads that you want to run. And so like in our case, we have these giant, I don't even remember what they call them. They're huge metal containers that then inside of them are the cylinders that will go into the actual germ-free isolator. They'll be connected at the port. And so we validate how many cylinders we can run at a time. Each one of the cylinders will have the biological indicators in them as well as the chemical indicators. And then the giant container that has more than one cylinder in it will also have biological and chemical indicators in it. So it's kind of a complicated question that, you know, it'll depend on the size of your autoclave and what you're planning to run in it.
Mm-hmm.
So I hope that answers
Okay.
the question.
Yeah. Otherwise, there's also, of course, the option to exchange more email communication. There are more questions. Very good. Another question is, could you elaborate more on mold traps and fire swabbing?
Yes. The mold traps are actually an interesting little method to do. Essentially, when the isolator is set up, an extra bag of feed will be put into the isolator, and that bag of feed is then dumped out into one of the cages. Then water is added. Obviously, the sterile water is added until the feed is all very moist. And at the time of setup, then, they stick into the feed pellets. Generally, it's 6 to 8 swabs so that the fuzzy end, you know, is down in the feed, and then the stick portion is out of the feed. And so then, excuse me again, every week they just pull out two of the swabs that are inside of that little mold trap. When we did the germ-free seminar, one of the participants also suggested, and I thought this was a fabulous idea, using LB media. LB media, Luria bertani, is just, you know, a broad general growth media, and they said that they take a wide mouth bottle of this media and set it inside of the isolator. And it performs the same function. Pretty much if a mold spore or a bacterial spore falls into that media, you're going to get turbidity. And so then you know you have a break in your isolator. And actually we're checking out this methodology at Taconic because, you know, it's simpler and it takes up less space in the isolator because, as anyone who's worked in an isolator knows, space is always at a premium. And having this extra box in there just takes up extra room.
Mm-hmm. Right. Next question, a bit longer. Why use dirty water when taking samples when you want to see if caging, feeders, and floor area are clean? Would you say if you have a positive anywhere, then things are done?
That's a good question. So what I mean by dirty water is just that the water is actually taken from water bottles inside of the isolator. And obviously, if anything is in that water, you want to know that too, because that does mean your isolator has broken and it's no longer germ-free. And the other portion of that question regarding if you have a break, are you through? It depends on your study. It really does, and what you are studying and what the purpose is of your germ-free isolator. At Taconic, we have many different designations for this, and without confusing everyone, we have our germ-free isolators, and then we have a classification for them, where we have a class 1 isolator, which means all that's in there. These are actually more of a defined flora isolator. So you know that those animals, say for instance, have been given altered Shadler flora, and that's all they have. So they have ASF and nothing else, and that's a class 1. If we have a break for mold, again, depending on the use of the animals, we may not discard it. We may continue to use that, particularly if the animals are destined, to go into a barrier ultimately or something like that. It really does depend on your purposes, and do you think that it's going to interfere with your studies? I mean, particularly mold, it is very, very, very challenging to keep mold out of an isolator. And I'm sure many of you out there who have done it know this. And it depends on your studies. It may not be a problem, and as long as you control properly and for the studies that you're doing, it may be perfectly okay. But if it's not, you are absolutely right. The isolator is done, and you have to start over.
Thank you, Paula. Next question. Do you ever use prepared stool kits to extract DNA from stool obtained from the isolator and then use PCR to determine if there is DNA there?
In general, we don't, not for our germ-free isolators. It is certainly possible. One of the things you have to be careful about if you take the molecular approach is that autoclaved feed will have bits of DNA in it, and these bits of DNA often do make it through the mouth, that is, into the fecal sample. And so even though there's nothing actually living in there, any bacteria, you could possibly get PCR products, which can cause you a lot of angst when there's really nothing alive in there. This is the reason that we tend to go with live culture, because we have had instances in other situations where we've had positive PCRs, but then found out later that it was actually the food that had bits of bacteria that had been autoclaved and shredded. So this can happen. So as long as you keep that in mind. And I think there was also an instance where we got some DNA out of, when we did that, the bedding itself. Sometimes you can also end up with DNA and then get a false positive. So just be careful with that. I'm not saying it's not a good approach, but there are caveats to be aware of.
Okay. There are more questions. This is very good today. Here is a question. Can the BBL port A cull transport tube be autoclaved and taken in?
Be autoclaved? Generally, no. And what we do, how we've been doing this, is we actually remove the sample from the isolator to a biological safety cabinet and put it into the transport containers. The transport systems that we've been looking at are the swab systems. And again, we collect the sample just as I described to you, and then remove it from the isolator, go to a biological safety cabinet, and make a sterile transfer. That way, you don't have to autoclave the transport system. These are transport systems that are used in human transport of samples, and they're all designed for human use. And they're individually sterilely wrapped, so As long as you open it in a biological safety cabinet, you should be good to go. Many of them contain a gel at the bottom, and so if you were to autoclave that, you would probably destroy the integrity of the media and also probably the nutrient components.
Mm-hmm. Thank you. I hope your voice still holds, Paula. So
Yeah.
another question. Do you do anything to assess for native viral contamination, such as norovirus and rhinoviruses?
Okay. For our germ-free isolators, it's all a matter of risk assessment and how you go about doing that. And our germ-free isolators are all from, of course, re-derived animals. And we do not test for any viruses. We do not do any health monitoring other than the vial testing. If we're doing a re-derivation for a customer, before the animals would ever leave the isolator, we would do health monitoring, and we would test for every virus and do our, if you're familiar with it, our IHMS 52, which is the most comprehensive health panel that we have. And so it would test for everything. But in general, no, we do not do viral testing.
Okay. Another question here. Do you use ATP-based tests for bacteria?
We have. We don't for our isolators, but we have done that for testing our cleaning procedures, our disinfection and sterilization of surfaces. Again, one of the issues we found with this was contamination, and it is very challenging to make sense of the data. We actually did this for some of our barrier testing and testing the sterility of the caging over time and the bacterial load on there, and the data were just honestly all over the place, no matter how well controlled we kept the experiments. So my experience with that system was not a positive one, and we ended up not even moving any further with it. Okay.
Okay. What is a wet mount test?
Okay, a wet mount is essentially you take a drop of that liquid. Let me back up for you. So recall I said that you take a swab, you go through the isolator, you add some feces, some bedding, a little bit of everything. So you have this nasty gamish of stuff. So you take a drop of that mixture, drop it on a slide, put a cover slip on it, and then look at it under the microscope and make sure that you don't see any bacteria in there or fungus or mold, because you will generally, if there is mold contamination, you will see the little hyphae, the little fuzzies in there. Sorry for the lack of technical terminology. I'm not a mycologist, but they're fuzzy and you can see them. And then the bacteria, you see the characteristic, rod shapes or cocci. And so we do that as part of our monitoring process. Okay?
Okay. Here's an interesting question. What if I plan to treat the animals with a drug or test article?
Okay. This sort of comes back to what we talked about with the irradiated feed, where you need to absolutely guarantee the sterility of your test article. If you're going to, give them a drug or a test article, you want to be certain that it is sterile. So what we do, not for germ-free because we don't do this, but for defined flora isolators, we will actually put a drop of the test article or the drug onto general growth media plates and do the same thing. It's a sterility test. You incubate it for 24 hours. If you don't see anything, you go to 48 and you make certain it's sterile. you know, again, it's a risk assessment. If it's part of your project, you have to do it.
Mm-hmm.
So you take the chance.
Okay, that was a lot of questions, and I think if there are no more questions coming up, well, there's one on the way. How are the swabs transported out?
They're inside of the vial. Recall I said
Okay,
that it all goes into that vial. So the sterile vial
in the
it
vial,
has a
right.
lid, so the swabs are inside of there.
I think here was a question, what kind of chemical disinfectant do you use for materials going into the isolator?
Okay, we generally use Expor or Clydox. Generally, though, it's Expor is what is sprayed when you attach a cylinder, you know, to the sleeve. Anytime you want to transport anything in, we have an SOP for how long things are sprayed, how long it has to sit, and then when you can open the little port.
Okay, so thank you everybody. That was a lot of very good questions we had today. I'd like to thank Paula for your interesting presentation and your very informative answers to the questions. I'd also like to thank all the attendees for the interest. Thanks for calling in. And as you can see on your screen, please look on our webpage with www.taconic.com for more webinars coming up. And in the course of our germ-free webinars, our next webinar will be on April 17th with the title Veterinary Care of Germ-Free Mice and Rats, a very interesting subject too. It will be presented by our Director of Veterinary Sciences, Dr. Tamara Good. And I look forward and hope to welcome you all again and even more people. And yes, see you again. Bye-bye.
Thank you. Bye.